This method will help researchers quantify microplastic loads in rivers, which often contain high sediment loads, as they are a major source of plastic debris to the ocean.
The main advantage of this technique is that it enables filtering and sorting of microplastics in water samples with high sediment loads at sizes not typically included in previous studies.
To begin, rinse the filtration device and nylon mesh sieves 3 times with deionized water. Then place the mesh sieves into each union joint, with the pore sizes decreasing from top to bottom. To prevent leaks, seal each joint tightly. Next, wet a mixed cellulose ester membrane filter with deionized water. While the filter is still damp, fold it into a cone. Then place a stainless steel mesh basket into the last union joint. Carefully place the folded membrane filter into the basket. Fold the lip of the filter over the edge of the joint. Next, place a mesh sieve on top of the membrane filter in the last union joint. Ensure that all of the union joints are sealed tightly. Then attach the hose from the top of the filtering flask to the base of the filtration device. Turn on the vacuum pump, ensuring that the pressure does not exceed 127 millimeters of mercury. Using a 500 milliliter graduated cylinder, measure the total volume of the sample. Next, record the volume of the sample and transfer it to the filtration device. To empty the filtering flask, turn off the pump and detach the 2 hoses from the flask. Then empty the flask into a waste container. To continue